Alright, so here we go with our second lecture on cell structures, part 2. So we talked about in part 1 basic cell features about size, membrane, cytoplasm, etc. And then we looked at domain bacteria and then the domain archaea and what makes them kind of unique and special and different from each other, different from eukaryotic cells, but also a few of the common similarities. So here we want to explore the eukaryotic cells. And so domain eukarya, the key feature for all members of that domain is that their cells have a nucleus. That's what we want to see, is that their cells all contain a nucleus. And if you have a nucleus in your cell, and then that nucleus contains your DNA, you go into the eukaryotic cell, or eukaryotic domain. There's also these little compartmentalization features of all the eukaryotes. We call them organelles. They're actually tiny little bags, if you want to call it that, that are structures, that are functional parts of the cell. They perform different tasks, they have different jobs, they do different things, and the concern is if those little organelles do not do their jobs, the cell can start to shut down and fail or doesn't work correctly. If a cell doesn't work correctly, in the case of a single-celled organism, it just dies. But in the case of something like the chimpanzee here, or the honeybees, or those flowers, the cell doesn't work, the tissue may not work. If the tissue doesn't work, the organ may not work, and this builds up to creating health problems. We'll talk about a disorder called Tay-Sachs. This is a genetic disorder that individuals can inherit. Mom gave them a piece of DNA, dad passed a piece of DNA to the child inherited 2 bad pieces of DNA, and one of their little cellular structures does not work, and sadly that child will die before they reach probably their 4th or 5th birthday. So we'll get into that in a little bit when we talk about cell structures. And then when we get into genetics, we're going to come back to Tay-Sachs and talk about cell structures in the genetic lectures later on in the semester. Okay, so a table to help you lock in all these cell structures. This is right out of the book, table 4.2, big red star tells you this is an important table to remember, to look over. It's got the key structures, nice little image of them, description, and then their function. I would highly encourage you to make sure you're comfortable with this. You're going to see questions on the quizzes and on the exams about cell structures, their functions, might be matching, might be multiple choice, fill in the blank, etc. But this is going to be a great reference for that information. Okay, so let's take a look at the general cell, the general eukaryotic animal cell. And we want to look at some of the basic features, the key components, and then we'll explore the plant cell, look at similarities, look at differences, just to get a feel for the general cell. There's lots of variations. We're just going to talk about big, broad generalizations here. So in the animal cell, the most noticeable structure, and if you look at an animal cell under a microscope, you should be able to see for this, the most notable structure is going to be the cell's nucleus. Okay, so the cell's nucleus is that big dark spot in the cell. Now, based upon the angle or the direction of the cell that you're looking at, you may or may not be able to see it. Cells are three-dimensional, but most of the time it's a very visible big dark structure there. Think about the nucleus like the brain center of the cell. The nucleus is a structure that contains all the DNA of a eukaryotic organism. So in a human cell, there's about 6 feet of DNA inside of that thing. And the DNA tends to be all strung out and kind of coiled up like a ball of string in this form we call chromatin. Inside of the nucleus is a darker ball. So it's like a dark ball inside of a dark ball. And that darker ball is called the nucleolus. This is the region of the nucleus in which ribosomal RNA is actually synthesized. So the chromatin is a genetic material, or we could call it DNA. It sits within fluid called a nucleoplasma, or plasm, and that nucleolus sits in that fluid, and then the whole thing is wrapped up in a membrane called the nuclear envelope. Now this is actually a phospholipid membrane. So the nuclear envelope contains all the DNA. And if you notice, there's a bunch of little holes, or we call them nuclear pores, within the envelope. These are just little tubes or holes within the nucleus, within that envelope, that allows information, or we would say that genetic information, a genetic code of RNA, to actually leave the nucleus. So we'll be getting into that later when we talk about cells and cell replication, DNA replication, and how that process works. But the key things to notice about the nucleus: you got a nuclear envelope, you have a nuclear pore, you have a nucleolus, and you have DNA in the form of chromatin all piled into that nucleus, and is all floating in liquid, or this fluid called a nucleoplasm. So generally, next to the nucleus are going to be these little structures called ribosomes. The ribosomes are little protein factories. This is where genetic information in the form of RNA gets moved. It goes between the ribosome, kind of slides in that groove there between large and small subunits, and then gets coded into actual proteins. So they're little protein factories. These are the sites of protein synthesis. And when we look at ribosomes and they're building proteins, they actually use different forms of RNA for this process. We'll see a ribosomal RNA, we'll see a messenger RNA, and we'll see a structure called transfer RNA. All of these are little helpers or assistants to the ribosome, enabling the ribosome to actually create a particular protein. Now, proteins are built from amino acids, but the code of RNA has to be read by the ribosome, and then it's actually translated into specific amino acids. And we'll come back to this when we get into genetics and DNA and molecular biology, but this is a key structure for proteins being built. And if you look at the diagram, the little picture there, the ribosomes are all those little red dots. Those red dots that are scattered throughout the cell. The bulk of the red dots are attached to a squiggly membrane right next to the actual nucleus of the cell. That membrane is known as the endoplasmic reticulum, or the ER. And most of the time, the ribosomes are embedded into it, making it look bumpy, so it's often called rough ER. So here's the image of the rough ER. All those little dots are ribosomes, and they tend to be closest to the nucleus. So information comes out of the nucleus, goes to the ribosome creates a protein. Now, outside of the rough ER, towards the outer edge of it, are those greenish tube-like structures. Those are known as smooth endoplasmic reticulum because they don't have ribosomes attached to them. The smooth endoplasmic reticulum is responsible for producing carbohydrates and lipids. So all these different lipids you're looking at on the left there, the fatty acids, glycerolipids, glycerophospholipids, etc., those are built in the smooth ER. So rough ER ribosomes make proteins, smooth ER makes lipids, and they can build some carbohydrates. They also can store some calcium. So these are your factories within the cell. These are the things that are producing products. So they are necessary to function correctly. If you're not making proteins, you're not making lipids, you're not making smooth carbohydrates, your cells are not going to be nearly as functional, and the cells tend not to work very well. So once we create a product within the cell, whether it's a protein, a lipid, or a carbohydrate, it often has to get packed into a bag. The bags within the cells are just generally known as vesicles. So a vesicle is just a little bag. It is made up of a phospholipid membrane, and then it has some type of content within it. Now the contents could be proteins, could be carbohydrates, could be lipids, could be lots of different things based upon the particular cell. Now the bags then need to get transported. They get moved from one location to another, and that requires the function or requires a shipping structure. So UPS is how you can ship a product. You put something in a box, give it to UPS, and they transport it around Springfield, they transport it around Illinois, to another country, all over the place. That's their role. That's their job, is transportation, to move things around. That is what these structures known as the Golgi bodies will do. So that squiggly stack of membranes there almost looks like a stack of pancakes. That is known as a Golgi body. You also will see the term Golgi apparatus, somewhat of an interchangeable term, but the Golgi is responsible for transporting stuff. It has one side of it that is on the inside. It faces interior to the cell. That is known as the cis face, CIS. Products come to it in these little bags called transport vesicles. They hit the Golgi. The Golgi takes it, blends it in, moves it through the Golgi, kind of rearranges it, reorganizes it. It might need to repackage it a little bit, and then it pushes it out the outside of the Golgi called the transface, and it puts it in a little bag known as a secretory vesicle. Now that bag may go out of the cell. If it's a hormone produced in one part of the body, it needs to go into the bloodstream and then transport throughout the whole body. So a simple example, testosterone in males is produced in the testes. Estrogen, progesterone in women is produced in the ovaries. Those hormones are put into little bags. The bags get packaged. The bags get pushed out of those cells. They go into the bloodstream, and then they're transported throughout the body through the bloodstream. Okay, so Golgis are responsible for modifying, kind of changing those packages a little bit, repackaging it, and then transporting it somewhere that it's needed throughout the rest of the cell. So Golgis, think about those like your UPS truck. Okay, so our next cell structure, or we'll call them organelles for part 2, is talking about the lysosome. Lysosomes are the cell's equivalent of the trash company, the garbage truck. They play essential roles in cleaning up cellular debris, breaking down waste, detoxifying solutions or substances, proteins, nucleic acids, pretty much anything. They can break down and recycle a mitochondria in about 10 days and get rid of it when the mitochondria is old and worn out. So these things are absolutely essential to cellular health and keeping the cells functioning correctly. So when we look at them, they're little mobile bags that cruise around the cell and move around, and when they find stuff they don't like, they attach to it. They really blend themselves into it, and then they secrete digestive enzymes and destroy it. So there's little bags full of these digestive enzymes that have the ability to destroy and detoxify things that they don't like. Whether it's cleaning up waste or destroying an invading pathogen, lysosomes are your garbage truck. We could also say they're your police force, your protectors. They are critical for your survival of your cell and the cleanliness. Now, earlier we mentioned just a little bit about Tay-Sachs. When lysosomes fail and they don't start breaking down glycolipids in nerve cells, the lipids build up, and that's not healthy. That causes the nerve cells to stop functioning. Your nerve cells decrease in function. That leads to the death of the nerve cell. That leads to the failure of the nervous system. And sadly, that young girl pictured there, she has Tay-Sachs. She would have died by the age of 3 or 4, maybe 4.5, due to complications of Tay-Sachs.